So thank you very much, Panthea, for inviting me to this seminar. So I'm Mélanie Prague. I'm a researcher at Bordeaux, and this work was also done by my PhD student, Marie-Alexandre, and all the data has been generated by Vaccine Research Institute and CEA. So please, during this seminar, you can definitely keep your question for the end, but if you really don't understand something or want more detail on something, please ask the question, and you can definitely also interrupt me. So today I will speak about a statistical pipeline that we developed in order to try to find mechanistic correlates of protection for SARS-CoV-2 after vaccination. Oh, I'm not sure. Okay. So let's start with a little bit of context. So why is it important? So as you know, there is an unprecedented worldwide effort to develop new vaccines. And as to date, there is still more than 350 vaccine candidates that are under development, even if the vaccine coverage in the world is up to today around 70%. However, there is still a real need for further development, and for many reasons, and three main reasons may be like waning immunities, the fact that we may still have the apparition of new variants of concerns, and also because we know that the vaccine efficacy in specific subpopulations, such as the immunocompromised population, is not that good. So we may still want to have some vaccine development for this disease. But the problem right now is that the comparison to placebo is going to be infeasible because first, it's probably going to be unethical, but also because there are too many people that are already vaccinated. Second, the comparison with other vaccine is probably going to be not significant. And finally, the clinical outcome will not be feasible at all because there is a very low incidence of COVID right now, which is probably good for everyone, but still not for clinical trials. So there is still a need to develop methods in order to find good surrogate marker for protection against COVID. And as per now, I mean, there are many articles out there in the literatures. I'll try to show surrogate markers and correlates of protection, but there is still some work that can be done here. And here we will try to address this question. And of course, the pipeline of statistical analysis we present here for protection against COVID could be extended to other infectious diseases. Okay, so let's start by talking about the surrogate. What is a surrogate marker or a surrogate outcome? So I will call S my surrogate marker, and it's an outcome for which knowing the effect of an intervention A, which will be a vaccine for me here, on the surrogate S, allows the prediction of the effect of the intervention on the clinical outcome of interest. So my clinical outcome of interest here will be infection by COVID. And so I'm trying to find a surrogate marker so that if I know the effect of the vaccination on the surrogate marker, it's exactly the same as knowing the effect of my vaccination on the fact of getting the infection. So this is kind of a well-defined concept, which dates back like 2010. And for example, we can take like the Prentice definition, which is the fact that if I want to find such marker, I need to find a marker that will first be correlated with the true outcome of interest. Second, this surrogate marker needs to be affected by the intervention. And last, the treatment and the outcome, so the vaccination and the fact of being infected, should be conditionally independent given the surrogate marker. So that's the pipeline, and that's what I'm going to try to find a method, a biostatistical method based on mechanistic model that allow me to check all these criteria. And so it's important to check all these criteria and to also keep in mind a certain number of problems when we try to define a correlate of protection. Here I listed 3 problems, but there are like obviously probably more, and most of them are kind of related with the causality. So first of all, as we all know, like correlation is not causation. And so we need to be like really meticulous when we try to find correlate of protection. So if I take, for example, an example in infectious diseases, and I'm looking at mother-to-child transmission of HIV, this is really related to HIV viral load. However, if we look at CD4, they are associated with the transmission. However, if I give, for example, an intervention, which is a IL-2, which is a type of interleukin that is supposed to increase the number of CD4, it will have no impact on the mother-to-child transmission. So it's not because I have all the correlation that I'm on a causal pathway, and I will really be able to show that an intervention is effective through a surrogate marker. Also, oops, sorry, I'm not sure why it did that. Also, so there is the fact that all the causal mechanisms, so in vaccination in particular, are really multidimensional. So for example, if we look at a study for pertussis, a certain number of vaccines, and basically if you only look at the antibodies titer, the IgG, it looks like the one leading to the biggest number of antibodies is the two-component vaccines. However, when the real clinical trial checking for efficacy has been done, they showed that actually the reduction of infection led by the two-component vaccine was significantly lower than the one led by the 5-component vaccine. And this is probably due to multifactorial reasons, but some reasons may also be that, for example, for the 5-component vaccines, they may be also not only a B cell, so antibodies response, but also like some T cells response. So there is really something multidimensional, and we need to address this problem by using methods that will try to take into account this multidimensionality of causal mechanism. And last but not least, in the method we try to develop, so we want to pay attention to the surrogate paradox with the off-target effect of intervention, so which is has been introduced by Van der Wiel in a biometrics paper. And it is that even if you have a surrogate marker, and you know that an intervention will reduce a surrogate marker, then it's not that your intervention will have the correct effect and an effect in the right direction on your true clinical endpoint. And for example, they looked at a myocardial infection, and they showed that actually the intervention tripled the death rate in this study. However, he had the correct effect on the surrogate. And for vaccination, it's really linked to something called antibodies-dependent enhancement, which is the fact that sometimes you may have a binding of virus to suboptimal antibodies, and actually, instead of preventing from infection, it will enhance the entry of the virus in host cell. So this is a concern that could happen in vaccination. Okay, so that's for the general concept on surrogate of protection. And when we go into specifics, into COVID, so there is a certain amount of work that has been done, and here is not at all meant to be exhaustive. But there was two meta-analyses that basically tried to relate like the neutralizing level with the protective efficacy. So in order to do that, what they've been doing is that they reported from clinical trials for many different vaccines, and you see it here, what were the results from the clinical studies. And so because all the studies were not in the same dimension, because it was all different biological measurements, they basically reported this as a fold change of a concentration of neutralization compared to convalescent sera. And so they reported that, and then they did a type of regression. And from that, they actually derived that the neutralization level could be seen as a correlate of protection, and that 50% protection against infection requires to be 20% of the mean convalescent level of neutralization. So this type of result has been mentioned and concerned like neutralization. But when you look at these results, so we can see that although it takes into account like the uncertainty that there is in all the clinical trials, it still have like a huge uncertainty. And also, like, it makes the hypothesis that the fold change is a good proxy because there is no standardized assay. But in fact, like, maybe there is a non-linearity in the biological measurement and something more complex could have been used. It's also only take into account one marker and not the multifactorial mechanism, and in particular the T cells. And also in itself, it's only look at correlation. It's not a mechanistic correlate of protection. And here I want to insist on the fact that we would like to be sure that we find a correlate of protection that is on the causal path to infection. Another thing that has been a lot used in the literature in order to investigate correlate of protection is the non-human primate studies. So this is very good because you can actually challenge the primates. So you know the exact dates and you know what the quantity of virus to challenge your primates with. So for example, there was this study from Corbett. And basically what they always look in these studies, and there was many more, is again, like the correlation between some immune markers related most of the time to neutralization and concentration of antibodies to the number of viruses and to infection. But again, they look at it most of the time looking at like regression. So this is still correlations. So this is what we will try to address here. And so as I mentioned before, we really want to have a mechanistic correlate of protection. So once you have a correlate of protection, it has been classified by Plotkin as either being like mechanistic or non-mechanistic. So if it's mechanistic, as I mentioned before, it has a mechanistic cause for the protection, which is to be opposed to the fact of not having a cause of protection, but predicts protection through partial correlation with another immune response. So here we want something mechanistic. And again, in some recent works that were kind of parallel to the one that I'm presenting here, Schiffer described like a nice pipeline of how having a mechanistic correlation of protection, and this passes by having a mathematical model behind that will describe the dynamics of infection. And this is exactly what we want to do. We want to use mechanistic model and a pipeline on mechanistic model to find a way to find correlate of protection. And why we want to use a mechanistic model, it's because there is this bunch of literature trying to show that mechanistic models that are in fact like nonlinear mixed effect model, they tend to very, very well estimate effects even when you have a problem of causality. And for example, there is this article that we've published like in 2016, I believe, in which we actually demonstrate that when you have an observational study and in which you know that you have indication bias, then if you use mechanistic model to model the dynamics, for example, of markers instead of a marginal structural model, then you can get correct estimate of treatment effect without having to use weighted approaches and approaches that adjust for the indication bias. So we really do believe that if instead of using regression and the classical framework of causality, you use mechanistic modeling, you could still really well handle this problem of causality, and that's why we decided to follow this path. So what we are going to do, so we are going to base everything on NHP studies, because first we had the chance to have the data for that, but also this is a very, very good preclinical model to study correlates of protection. And thanks to mechanistic model, we will try to do causal inference to go beyond correlation and define mechanistic correlate of protection using model-based approach. And then we will apply this framework to various studies. Okay, so let's move to the methods. So in order not to start with like all the specific methods details, I will just give you an overview of the study so that you really understand how I frame my problem and how are my data. So I'm in a preclinical study in non-human primates. So all my non-human primates has been vaccinated. And then after 4 weeks, they get challenged with SARS-CoV-2. And basically, I have 3 groups of treatment in this specific study. I have a group of naive monkeys for which they never had the infection before. One group of convalescent monkeys for which they had the infection before, between 26 and 24 weeks before the challenge. And finally, one group which was convalescent and vaccinated. So those are the 3 groups. And basically what is done is that after the challenge, these monkeys are followed up almost every day, and every day there is a certain number of markers that are collected. And the one that will be of most interest for me, and among which I will try to find information, are the viral load. So the viral load being the number of viruses in my monkey, which is basically a proxy of the infection. So is there an infection? And also if there is an infection, how strong it was. And I will try to browse for correlate of protection among the baseline covariates that has been measured, and that I will assume and I will test also if they are related to the vaccination. And among those covariates, I will go further in detail after, but I had cells counts with intracellular seed cytokine stainings. I also had the seed cytokines. I also had some neutralization antibodies, and I also have some measurement of the quantification of antibodies. So at the end of the day, I was trying to browse a correlate of protection among something like 70 markers. Okay, so if we look at the data now, first at the viral load, because as I said, this will be kind of my proxy, this will be my outcome that I try to control. So here are the viral loads that have been observed in my monkey. And these viral loads are observed both in 2 places in the monkey, in the nasopharynx and in the trachea. And you can see that the dynamics seems different from one group to another, with a higher peak and a longer infection in naive than it is in convalescent and than it is in the vaccinated group. So once I have that, what I said is that I want to use a mechanistic model to model this data. And there is a large number of models that are out there, and modeling viral dynamics is like a huge field. And what we've been doing here is just using a classical target cell limited model. So in which we assume that we have 3 types of cells for the immune system. We have like uninfected target cells that can be eventually infected by the virus, the infectious virus, and it turns that into an infected cells at rate beta. And then those infected cells, there is an eclipse phase until they become productively infected cells and they produce viruses. So how we extended the classical model of target cell limited models in this work is that basically first we added the vaccination. So we added a specific compartment which is linked to the vaccination, which is kind of an input at baseline. And also we modeled separately nasopharynx and trachea. So this is a mechanistic model. And so what I need to have a population approach on mechanistic model, because if you get back to the curve, I mean, there is not only one curve. You can see that there is a huge variability in this observation. And this variability can be due to inter-individual variability or maybe also error measurement. So my mechanistic model is defined as follows. So there is a system of differential equation which describes the system that I have here, and for which I explained a little bit like what is the framework. Then I suppose that this system is basically the dynamics are led by the parameters of the system, like infectivity, death rate of cells, and production rates of cells. But this parameter, I will suppose that they actually do follow a mixed effect model. So they have a mean value in population, which can be eventually depending on covariates. And you will see that some of the covariates will be the group of vaccination, for example. And then I have a random effect that models that every individual is specific. So it's statistically constrained by a normal law. But again, every individual will have a slightly different value from the population value. And finally, I have an observation model. So this observational model is first here to take into account the fact that I'm not observing all the compartment of my system. I'm actually only observing what I have in blood, in the trachea and in the nasopharynx, which is the number of viruses. And also I observe it with an error measurement, which just reflects like imperfect measurements. So this is my mechanistic model from which I will try to analyze the data and build the pipeline to find the correlate of protections. So as I mentioned, like a correlate of protection is a surrogate marker. So here on the upper right corner, you have like the small DAG that I showed at the very beginning. And so we need to check that all the arrows with this mechanistic model that I just described, all these arrows do exist, and have the right properties. Okay, so first we need to see if for the vaccine, there is an effect on the outcome. So the outcome being like the viral load. And this effect will be mediated to the fact that the vaccine, so the group, will actually have an effect on the parameter of my model. So as I mentioned, I have this mixed effect model, and the parameter of the model which lead to the dynamics of the markers and the viral load specifically may depend on covariates. And so first, I want to test this if there is a signal here. So I want to test if on some parameter, there is a group effect. And if it exists, then I will be able to follow up and say, okay, now I want to find a surrogate marker for this group effect. So that's the first step. Then once I have done this first step, I will try to find a surrogate marker. So the first thing is that this surrogate marker needs also to be associated with the outcome. So basically, I can, for all the markers that I'm thinking they could be correlates of protection, I could do exactly the same analysis and replace one by one this covariate effect by the marker in question. And so that will provide me with many models, and all these models, I will be able to compare them using a criterion. So I could decide to use AIC or BIC or BICC, whatever. I just have to stick to one criteria. And then I will be first able to kind of screen all my markers one by one and see if there is one marker that actually first lead to one, the best AIC, so the lowest in the case of AIC. And also I will somehow be able to see if adjusting for this marker is actually leading to a better model than adjusting for the group effect. And if that's the case, then this is the first hint that maybe, I mean, one of these markers, and especially the best one, could be this surrogate marker I'm looking for. But as we mentioned before, this is not enough. And actually, what we want, we say that we want that conditionally on this marker, we want that the outcome and the intervention become independent. And so this is possible also to test here with a statistical test. So basically, I can compare my model in which I have just the effect of group on my marker with a model in which I have both the effect of group and the effect of my marker. And if it's really a surrogate of protection, then what I should actually expect is that whereas my group effect was significant in my model in which I had only the group effect, it should become non-significant to the profit of the other marker if we are actually looking for a surrogate marker that has the good properties. And finally, then what I want also is to check that actually like the intervention has an effect on my surrogate marker. And so this could be checked by just correlation, of course, but one way to check it, we believe in our mechanistic model, is to look at the magnitude of the variance of the random effect. So basically this term, the random effect here, will quantify the residual heterogeneity that I have on my marker once I adjust for the group. And if I believe that the marker is actually a marker that has a bit that is influenced by the group and that has a better information than the group, then what we think is that the magnitude of the variance of the random effect associated in the model should be lower when I adjust for the marker than when I adjust for the group. So the magnitude of the residual heterogeneity is better captured with the marker. So those are basically all the pipelines that we would like to apply. So it's based on mechanistic models. So we hope it handles like causality properly. And we also check, thanks to those three criteria, all the arrows of the DAG that needs to be there in order to properly define a surrogate of protection. So now let's see how it goes on the data. So basically, we just like took this model. So here are the differential equation if I write them down. So it's just like showing transfer rates between the different types of populations that I have in my model. So as I said, so we need to find on which parameter we have an effect of the group. So this is the topic of some very nice work we try to do here on how do you do automatic selection on effect of covariate on parameter in mechanistic model. So we used like some algorithm like that here, but this is not the topic of the talk. So we ended up finding a group effect on beta and delta. And we had an observation model linking what we actually observe in my monkey, so the viral load, with what are the compartments of my model. So we had like to solve a certain number of issues that are probably not relevant for this group, but I mean, there is some parameters that need to be fixed, the inoculum, the number of target cells, also like the volume of distribution. Like there is a certain number of parameters that had to be fixed and relates to biological assumption that we had to do in order to have a system which is identifiable. We also tested, like, as I said, like one of the specificity was that we modeled like the nasopharynx and the trachea with some exchange rates. So we actually tested if we could identify like what was the rate of transfer, and this was basically not very conclusive. So we ended up like modeling the nasopharynx and the trachea independently. And finally, for some parameter, there is an identifiability which is not complete. And so instead of doing like regular estimation, we perform like profile likelihood on which we have a grid search and we try to find like values that provides the maximum likelihood, but we are not doing like proper estimation. At the end of the day, for the other parameter, we did do, so all the parameters that are here in the like 3 first block, we did perform like a regular estimation. Estimation is based with an SIEM algorithm as implemented in Monolix, which is from Lixoft company. And what's important here is that we managed to show that there is a significant effect. So we managed to show this red dot here, like on the data I'm considering. So we could see on the graph that it seems to be an effect of vaccination on those dynamics, but it's actually confirmed by estimation and statistics with significant fold change of some parameters in the real data. So those are the feeds, just if you on few monkeys, just if you want to convince yourself that, I mean, the feeds, I mean, the data are very sparse and noisy whatsoever, but the feeds are not that bad. And those are visual predictive check if you want to have like a bigger overview, not just to see monkey by monkey, but just how good is the fit. So basically, it's kind of satisfying. And so at the end of the day, so we managed to show this, and just if you're interested in this vaccine, so there is a strong effect on viral infectivity with a reduction of 80% of viral infectivity by natural immunity and by 99% by the vaccine-induced immunity. And there is an additional effect on the death rate of infected cells, which could be mediated by a T-cell response. I mean, this is possible that this is largely due to B-cell response, and this is largely due to T-cell response. Okay, so now let's browse into my potential correlate of protection. So here is a list of all the correlate of protection that I'm going to test. And here I'm going to test like if there is an association between those potential marker and my outcome of interest. So to do that, as I said, we just like compared for all the markers that I had available in the studies. We fitted like a separate model. And here what we wanted to compare is the AIC. So in green, you have the AIC for the model adjusting for the group here with the line. And you could see that we managed to exhibit like 2 markers, and the one RBD-SE2 is probably the one which is the most like promising if we trust a difference of 5 points on the BICC, but this is a different story. And so this is the one that we decided to push further in order to investigate if it is possibly a real correlate of protection or not. And so the fact is that we checked that. So basically, we adjusted only for group, and then we could see that all the covariates were statistically significant. So the group covariate was statistically significant. But when adjusting for the group and the marker, then it became non-significant at the profit of the marker. So it checks like this conditional independence that is needed. And finally, for the third criteria, 65% of the variability is explained by the group, and 87% of the variability is explained by the marker. So again, there is a hint that there is a bit, the marker better capture the relation, better captures the heterogeneity than the group. So at the end of the day, what we concluded is that this marker, which is a marker of neutralization, so it's really like measuring functionality of the antibodies, is probably the most reliable correlate of protection that we have in this study. And this is really, or at least we believe, a mechanistic correlate of protection. So we can really say that this is on the causal pathway. So then we could say, okay, but now we can do a second run. So like once I have adjusted for this marker, like which is my first correlate of protection, we could try to see if we can adjust simultaneously on the second marker and like do kind of a 2-step things, like try again to see if there is another marker that can enter the model, whereas there is already the ACU2 RBD binding inhibition in the model. And here you can see that when we look at the BICC of the model with one additional potential variable inside, I mean, it's definitively like not super conclusive. And in order to, and even if you take like this guy, the interferon gamma here, and you try to add it to the model, it whatsoever doesn't check the criterion 2 and criterion 3. So at the end of the day, we really decided to keep only one marker in the model and say that the correlate of protection we found is really this marker. And so what's interesting is that these results that I showed you here on some specific data is also validated, and you can check in these papers are all the follow-up results and all the results for the other study, but it's been found also to be the same marker that is relevant in those two data. So the first one is another data on, the first one is a data for mRNA, and the second one is another vaccine that has been done in the same platform as we do. So we had the exact same biological measurement in terms of units and also in terms of mechanics. And so what's interesting, and just, I mean, I'm almost done with that, but I think one of the very interesting feature on top of like finding correlate of protection is the next question after saying, okay, it's interesting to look at this marker specifically. The next question could be, okay, but is there a threshold that should be reached by the marker? And the fact that we use mechanistic model also allow to some extent, and we are still digging on this question, but to answer this type of question of finding a threshold on a marker. So because we are using a mechanistic model, we can compute, and it's a viral dynamics model, so it's like epidemics dynamics, we can compute the basic reproductive number, which is basically the number of infected cells produced by one infected cell, so same as in epidemics, and that as a function of the parameter of the model, among which like beta, for example, is a function of my correlate of protection. And so from that, I can like look at each time point during my study, what is the effective reproductive number of all my monkeys? And then from that, I can just like take at every time point, like the estimated R0 as a function of my marker of interest. So here it's like neutralizing antibodies. And I can basically have this type of scatter plots. And from this type of scatter plots that I really generated through the mechanistic model, then I can basically like fit, so for example, here we just did like a simple polynomial regression, but from this polynomial regression, we could basically infer like a lower bound for which we predict that if the marker, so neutralizing antibody, is higher than this lower bound, then there is 95% of chance that the R0 will be lower than 1, which means that there will be no infection. So we can like kind of reconstruct a bound from the data and the search that we've been doing. And I think that's a very nice additional feature. And there is probably more clever way to do it than just a simple polynomial regression. Maybe in one integrated approach, in a one-step approach. So far we are still like digging this question, but I think it's a really nice feature. Okay, so as maybe a conclusion, so we have proposed a modeling approach to explore the mechanistic effects of vaccine and assess the quality of markers as mechanistic of correlate of protection in NHP studies. So we have shown that the blockage of new infection of target cells is the main mechanism of vaccine studied at least studied here. And it's also the main mechanism that we found in the natural infection. And we've shown that this is well captured by the neutralizing assays, so which we found as being the correlate of protection. And we confirmed that on 2 other NHP external studies. And we also shown that, I mean, we could have like some additional mechanism, especially on the infected cell death rate, the death rate of infected cells. But this was a way less strong signal. And that's why we didn't qualify like any of these T cell response as being like a correlate of protection, and we just kept like the neutralizing assay as being a correlate of protection. What we didn't do, like probably a lot, but one of the very main limitation, I think, to this work is the fact that the antibodies and all the markers that are considering as a surrogate marker or correlate of protection are actually measured probably with errors. And this type of error we do not take into account at all, because we assume that the value that has been measured is the true value that entered my model. So that's the first thing. And the second thing is that there is an influence of the viral dynamics on the creation of antibodies. So even in my infected monkeys, they eventually get a little bit the infection. And so they are in contact with some virus produced de novo. And so it means that this will also stimulate their production of antibodies. And there is really like a feedback loop here that could be of importance. And that is not properly taken into account in the mechanistic model we've been building so far. And finally, I talk a lot about the T cell response, which is probably like kind of mediated by the death rate of the infected cells, because CD8, for example, will probably act on this parameter. So this is taken into account in this model, but we could think about extending the model to have like a proper pool of effector cells that is actually exactly modeling the T cell response, and like have a more complex mechanistic model to better try to model this causal and multidimensional mechanism. So this is where we stand in terms also of direction. So we have tried to complexify the model and end up like in one integrated approach, in which we also try to analyze and estimate the dynamics of the antibody to correct for the error measurement we may have, and the feedback loop between antibodies concentration and viral dynamics. But like, yeah, it's still under progress. And yeah, and I'm happy to have like any feedback on any limitation or things that we could improve in this approach. And I just would like to finish by acknowledging like all my collaborators, and in particular Vaccine Research Institute with Levy and CER with Roger Legrand, who generated all the data that we've been using in order to be able to have a very nice illustration of this method, and also hopefully like provide some new insight on correlate of protection for COVID. And yeah, I'm happy to take any question. And also if you want to contact me by email later, I'm happy to discuss further. Can you hear me?
Yeah, we can hear you. So thank you.
Okay. I was like, okay, maybe they ask a lot of questions and I could hear them. Okay.
Thank you very much for this nice talk. We have some time for questions, so if anyone from the audience has a question, please unmute yourself and step in to ask. perhaps I can make a start and question whether you have sort of tried to understand what the power is in those type of studies, because you've mentioned that you have like possibly quite a few of these markers and noisy data, and perhaps the signal is not so strong for some of them. So is there any simulation that you have done, or
Yeah, so oops, I don't know why you did that. Yeah, so there are two things. So first, in terms of calibration of studies, so we actually did work with a vaccine research institute in order to design the preclinical study. So most of the time what we use is PEFIM. It's based on the Fisher information matrix, and it's specially designed when you know you are gonna analyze your data with a mechanistic model, then and you want to show the effect of group. So this was calibrated to show the effect of group. Then we made sure that we could have a sufficient power with those 18 monkeys to detect the effect of group. So we know we are powerful enough in this NHP study, which is low sample, but it's always the case in preclinical study, to detect this. And second, we had this concern, so here, of kind of multiple testing, because, I mean, we are not doing multiple testing because there is no test. But still, I mean, we are calculating like so many AIC that we wanted to make sure that if we just like create 70 random covariates, like just noise, there will not be one that is accidentally better in terms of BICC. And this was basically to validate the fact that when we see like a model that is really better, it's really because there is a signal. And I believe this is in the supplementary material of the paper. So we did this simulation. And so when we have only noise as a covariate, then there is like no signal in the BICC. So in this figure, there is no signal. So those are the two things.
Thank you. Thank you. Is there any other question from someone from the audience? I have more, but perhaps I'll give the opportunity in case someone wants to ask. You also mentioned the problem of measurement error in some of your biomarkers. I mean, is that purely a measurement error in the sense that it just reduces your ability to identify, or is there more systematic, like time dynamics that it could?
The way we model it is assuming that it's pure noise. Mm-hmm. It's the noise we need to get rid of to really access the true trajectories and be able to be more precise, but it's just a random noise. And what we've been doing also is having some proportional errors. So that can often happen, like the magnitude of the noise is bigger when the size of the marker is bigger. That happens most of the time in pharmacometrics. So if you have a concentration from 0, it's probably like small error, and a concentration very high, it's like bigger error. So that we tried, and it doesn't change the result much, but we didn't try any type of systematic error measurement. Also because it was not something raised by biologists. So yeah, but I'm not sure how it will behave if it's the case.
And yeah, I have one last one, and it relates to the identifiability issue that you mentioned at the beginning. I mean, of course, like, it's very complicated to run sensitivity analysis of these things, but if, say, someone wanted to ask you, like, which ones of these assumptions that you made are the most influential, and follow up on them, is there a way to do it?
Yeah, so first of all, what we try is to check like the mathematical identifiability. So once I have a model like this one, I use DAISY, which we use DAISY, but there are probably many methods out there. for which you can test algebraically if it's mathematically identifiable. But this assumes that it's identifiable without error of measurement with a process that is measured continuously. So we always check that, but the problem is that like that's my process. So I mean, it's not continuous at all. It's unsolved sometimes also. And on top of that, like it's probably full of error measurements. And so what we face is more something like practical identifiability. And in order to check that in our data, we still have information. So we do sensitivity analysis a lot. And also we do profile likelihood. So before trying to estimate all the parameters all together, we try like we fix one to different value and we check that for different value, it leads to different likelihood. So that, I mean, we know there is practical information and we can believe that, I mean, when we will do the maximum likelihood at the end, it's actually capturing a maximum and not just wandering on a flat surface. Yeah.
Thank you. That answers my question. And I think we have time for one more question from the audience. If there is any, please raise your hand or unmute yourselves.
And I forgot to mention also that we have a lot of postdoc positions open. So we never know. I forgot to tell it at the end, but in the team, like we do have postdoc position open to work on this type of mechanistic modeling and extension of this work or something slightly different on modeling. So like, please feel free to contact me if any interest.
Thank you very much. And so we'll thank you again for a very nice talk. And for letting us know about these opportunities. Looking forward to the follow-up that you mentioned in the end. And yeah, many thanks again for the talk. It will be recorded and uploaded to our YouTube channel, so you can share with colleagues if.
Okay.
Yeah.
Thank you so much.
Thank you everyone for joining. Bye.
Thank you.